Okay, I changed the circuit because until now we have heard quite a lot about the hippocampus. Now I move to another part of the brain, which is the cerebellum. And this part of the brain has a general organization that has some resemblance to the hippocampus, so some concepts are the same. The neurons are always neurons. So what is different is the organization of these neurons and the specific properties of the neurons, which causes some change also in the way we have to conceive the experiments and the models about the cerebellum. I would like first to start with a general introduction on the conceptual framework, which I think is very useful at this point. Why do we need models? We need models because we wanted to understand. We normally make models spontaneously in our mind. We reconstruct the models of the world continuously, and if, to be even more clear, our brain is a sort of machine that makes models. Models of the reality, models of what we think, models of whatever. And then these models are compared to the real world and used to live in this world. So making models is something that is quite spontaneous for us. The problem is how to make them. In the case of the brain, we have to face a lot of serious problems. The brain poses multidimensional issues. For example, we have many parameters to compare and to correlate one with each other. The complexity is very high, which means that the number of interactions between these parameters is very high. There is a problem of scaling because we have different scales at which the brain is organized, from subcellular to cellular, neurons, microcircuits, large-scale circuits, and then the brain. And from another point of view, we can see the brain as organized in structure, function, and dynamics. The anatomists look at structure, the physiologists look at function, but I think that all of us should look at dynamics, because what drives the final operations of the brain is dynamics, which means the development of activity in time and space. And until we do not understand dynamics, we don't understand the brain. So we can make guesses starting from function or starting from structure, but we can't understand until we get dynamics. And to understand dynamics, we need models, because it is not easy to reconstruct these dynamics. Another problem which is critical is that what we understand from the brain in humans or in animals comes from the observation of facts, or observation of activities, which means that we take ensemble signals like the fMRI signals, the electroencephalographic signals, or whatever of that kind, and these signals don't tell anything about the single neurons. They just speak about the ensemble of neurons working all together. And now we have to face a problem which is rather nasty, is what is called in mathematics an inverse problem, which is also ill-posed. This problem cannot be solved easily because we have many solutions to the problem. You can imagine that to obtain an fMRI signal or an electroencephalographic signal, you can have different arrangements of the neurons working underneath. You do not have a unique solution, except if you have a good model. If the model is correct, you can have the unique solution to the problem. So there are several issues that have to be considered, and the original conception of a human brain project was just done to cope with this set of issues. To go down to the very basic facts, we have the data and we have the models. So we have to use the data to make models, and then the models will tell us what data are missing and what we have to do in the experiments to look for something that is missing. We have holes in our knowledge. We don't know everything. So we have to use the models to understand the data and to request new data from the experimentalists. Only at that point we can generate meaningful brain theories, which is our target. Understanding the brain means having a theory, not having a model. But to understand the brain, we need the data and the models. So, I mean, this is something that has to be conceptually framed properly. Otherwise, we don't get into the issue. What means making models? Making models means that we have this biological data on the multi-scale, and this biological data can be used to reconstruct models. What I'm telling you is that I'm going toward a bottom-up approach. It's the one that has been explained by all of us in these days, which means we start from bottom and we go up. We start from the molecules, we go to the neurons. From the neurons, we go to the microcircuits, and then hopefully to large-scale circuits and the brain. We have to pass through steps of construction, validation, and propagation. Construction, you have seen a lot about constructions of models. Validation means that we have to confront the outcome of these models with data that have not been used for construction. So we have to predict. So the model has to predict functional states. And then we have to propagate the model into networks in order to have it running and working, contributing to ensemble activities. What happens next? What happens next is that with this kind of realistic models, we can't go that far. We get stuck into the microcircuits basically at the moment due to the complexity of managing the models, running the models, the availability of supercomputing resources. So what we can do now is to try to simplify the models in a way that we maintain the salient properties. We simplify while we don't lose too much. It's a delicate step. After we do it, we can make networks that grow in scale, becomes big, can be interconnected. We can make real reconstruction of brains or circuits of the brain. And then we can embed all these networks into control systems or make real virtual brains that run into robots. And these robots can interact with the environment. So the robot is an artifact that interacts with the environment. And in that way, we will see our molecules, our neurons, our networks that run and interact with the environment. You would say, why we need to do that? Can't we put an electrode in the brain of an animal and then look what the neurons are doing? Well, in that case, the experiment is difficult. There are a lot of ethical problems. We do not see much more than one, two, or a couple of neurons, and we never know exactly what we see. So we carry out a very nice and very useful observation, but it is largely uncontrolled. We don't know exactly what happens inside. We just pick up some element from the whole system, and then we have to try to infer what this element was doing in what circuit and what's happening. So basically, this is an exceptional way that allows us to understand what the neurons are doing when a brain is interacting with the environment. We can make an hypothesis of what is happening. We can go to theoretical models. We can request for more biological data. So this is the loop that we're going through. This is just to be clear. So this is the basis of all the story. Then we make the models. The models that we've seen in these days are basically systems of ordinary differential equations that are more or less complex depending on the kind of neuron that we want to simulate. And this is a Hodgkin-Huxley style representation of what the neuron can do in the membrane. And this representation is based on the determination of biological parameters, mostly these conductances. These conductances are the critical parameters. We have seen about it already, so I don't go back on this point. We can do the same work with the synapses. The synapses can be represented dynamically as a cycles of a neurotransmitter release, recovery, and release again. So these cycles are again differential equations. So we are in a system, in large systems of differential equations. A neuron can be represented by tens of differential equations, or even hundreds or thousands. I will show you an example that was explained by Stefano yesterday. The Purkinje cell has about 65,000 differential equations. It's huge. So the problem is that with all these differential equations, we can't get that far with the supercomputers that we have now. So we have to simplify the neurons. In the simplified neurons, we can have one differential equation, two, three, a very small number of differential equations. But still, we can maintain some of the salient properties of the neuron, some, not all. Again, this is the last general picture. Then I go to the cerebellum. We start from biological recordings, and then we can construct the realistic neuron models. And these realistic neuron models can be used to generate detailed neuronal microcircuits, and we can test the internal dynamics of the microcircuits or their input-output functions. That has been shown, for example, for the hippocampus right now. What we can get is information about network biophysics and computation theory. And we stop there. The other way is to simplify the neurons, generate simplified networks, simplified brain models, embody these models into the neuro-robots. And here we have another interesting point. The networks do not work efficiently unless they have plasticity inside. Plasticity has not been mentioned very much during these 2 days, but plasticity is critical. Plasticity gives the weight to the synapses, which means that it regulates the communication between neurons. And the regulation of this communication means that we can switch off a synapse, switch on a synapse, or calibrate the synapse, which changes completely the way a network is working. Completely. Can be switched off completely, turned on, or put in different computational states. And if we do not have plasticity in the network is half dead. So how to put plasticity into these networks? Clearly, we can put it into the realistic, complex representations of the networks that we are seeing now, or we can put it into simplified networks, which is much easier. So we can have networks which are simplified but have plasticity. And having plasticity is not just a question of putting it into the network. Having plasticity means that in order to have it working, we need to iterate activity in the network many, many times, which means long cycles of simulation. You can imagine running the network that we've seen for the hippocampus for 1 hour to allow plasticity to develop. It is impossible. It's really impossible at the moment. So in order to get, I can ask Armando to have 1 second of activity of your network, how much time do you need on a supercomputer? 3 or 4 hours. 3 or 4 hours for 1 second. 42,000 cores. So this gives you the scale of the problem. And the same is for the cerebellum, where I will go very quickly. So you can imagine running for 3,600 seconds, which is 1 hour. Becomes unfeasible. So in order to test the impact of plasticity, we need simplified circuits at the moment. This is the best thing we can do. And therefore, coming to this point is not just, let's say, an exercise for informatics, but it is very important in order to reconstruct the fundamental property of the brain, which means having networks interconnected with plasticity. From this point onward, if we are clever enough, we can put all the stuff into a neuro-robot, carry out closed-loop testing, and simulate behavior and go up to control theory. We have elaborated the whole loop with the cerebellum. I come to the cerebellum. The cerebellum is very useful in this sense because the cerebellum is tractable in terms of circuitry, much more than other parts of the brain, because the cerebellum has at the back a very strong theory on what it should do. We always have to consider the theory. For the hippocampus, we have the theory that the hippocampus is important for memory, for learning and memory. Not all kinds, but some kinds, okay? To look at the memory of the hippocampus, you can't study the hippocampus alone. You have to study the hippocampus with the rest of the circuit that makes it able to process memory at the behavioral level. Otherwise, it remains a microcircuit, like the cerebellum would remain a microcircuit. The cerebellum doesn't process movement or cognition as it does if it stays alone. It processes movement and cognition if I connect it to the rest of the brain. So in order to get up to the level that we would like to achieve, which means understanding what the microcircuit is doing, we have to reconnect the microcircuit. There is no way. And to do it at the moment, we need to pass through the simplified models. I think these concepts have to be very clear. I mean, I'm not sure everybody, also at high levels, understands it. So now, this is what happens. We have started from neurons. We have done realistic models. We have simplified the models, embedded the models, run the robot. And this is a Purkinje neuron of the cerebellum that is learning to cope with a task. Is learning to cope with the eye blink classical conditioning, which is the simplest task that the cerebellum controls. It's a monodimensional task. It's all in time. There is no space in this particular task. Then we can scale up the problem and go to more complex tasks, which are bidimensional, tridimensional, multidimensional, which is what normally the cerebellum does. It copes with thousands of parameters and binds them together into a single output, which is the control of timing of a movement or control of timing of a thought. So this is what happens in the deep cerebellar nuclear cells. When the Purkinje neuron is switched off, the deep cerebellar nuclear neurons fire very precisely with millisecond precision. This millisecond precision is the precision that we need to control movement. Our movement is controlled on the millisecond time scale, and this comes out from emerging properties of the system. This is the kind of information that we get out from this process of simulating, starting from the data, going through the realistic models, simplification, and then embedding and closed-loop testing. Okay. I will show something more during the talk. The cerebellum is a network that is made of neurons. Some interesting informations are that the cerebellum contains half of the neurons of the whole brain. In the mouse, we have 65 million neurons. And of these 65 millions, 35 millions are in the cerebellum. So the cerebellum is not a secondary structure that does some funny operation in the brain. The cerebellum contains a lot of neurons here in the granular layer, then has Golgi cells that are the inhibitory neurons, and these Golgi cells control the granule cells. This is the input of the cerebellum. Then there are Purkinje neurons that are the most famous, which are the principal neurons of the cerebellar cortex. The Purkinje neurons are also the biggest neurons of the brain. These neurons can receive up to 1 million synapses in humans. This is an incredible number. There is no comparison in any other kind of neuron in the brain. They're huge. In the mouse, we have about 200,000, which is already a big number. And then there are other neurons that control the Purkinje cells, which are called the molecular layer interneurons. They're inhibitor interneurons divided into two classes, stellate cells and basket cells. The way all these neurons are connected and wired up makes a lot of problems in terms of reconstruction of the circuit, because they have a strong geometrical organization. They are not isotropic. They have specific organization of their processes in space. They go on one plane, they don't go on the other, they cross in one direction, they don't travel in the other, and so forth, generating a geometry that was early recognized to be quasi-crystalline, and it was called a lattice. This lattice is an organization that is very different from that of the hippocampus and from that of the cerebral cortex. So, or you can begin to understand that using the same tools that we use for the hippocampus and the cerebral cortex to reconstruct the cerebellum has to be treated very carefully. So if we can use the same tools if they are very general. And if they're very general, they will be valid for everything. If they're a bit too specific or bent toward a specific property of the network, we can use for one and not the other. The output of the Purkinje neurons go to the deep cerebellar nuclei, which are another mysterious structure, because everybody speaks about these nuclei as if they were made of one single neuron that stays there, takes an input, and sends out an output. That's clearly not the case. These nuclei are made of at least 5 or 6 types of different kinds of neurons, which are interconnected in a way that nobody knows. So the output of the cerebellum is still a mystery. And what happens after this output? The signals go back to the cerebral cortex, so they close the loop with the cerebral cortex, passing through the thalamus. They go to the brainstem, to several nuclei in the brainstem, where they control the descending and ascending pathways. So every kind of signal that goes out from the cerebral cortex or up to the cerebral cortex is eventually controlled by the cerebellum. So the cerebellum is embedded in loops. And this tells us how to wire it in the brain of a robot, because this fundamental anatomy is known. What is the main function of the cerebellum? It can be summarized in a single sentence. The cerebellum allows to predict the precise timing of events, of correlated events. Predict means prediction. It is the so-called prediction imperative of brain function. It means the brain predicts, doesn't react, predicts. Can also react, but that's the low level of the brain. The higher part of the brain predicts. The timing is a timing machine. And I've showed you already that the precision of the cerebellum can scale up to the millisecond, while the precision of the cortex is around 100 milliseconds. The cerebellum is between 10 and 100 times faster than the cerebral cortex. So the cerebellum is fast and helps to predict timing of correlated actions, which means can put the different components of an action or the different components of a thought in the proper order in time. And by being distributed, since the computation is distributed in space, it can also control the distribution in space of the different components of a complex problem. This complex problem can be cognitive, purely cognitive, or cognitive applied to movement. Again, be careful. Motion is cognition. Motion is the extension of cognition into movement. Okay? So don't be surprised when I tell you that the cerebellum is not only for movement, but is for cognition. 80% of connections of the cerebellum in humans do not go to the motor system, go to the associative system, which has nothing doing with motion directly, but is designed to control our output, which is motion. Okay. So the cerebellum is a very special machine that is wired in this way and then controls several functions of the brain. So now I skip some details. I want just to tell you that the story started with these guys. For example, David Marr is one of the most famous. Albus and Ito, other scientists that have given their activity into the field of the cerebellum. And they have discovered and understood by mixing up sort of experimental, theoretical, and modeling approaches that the cerebellum has to work in this way according to their theory. It has to receive inputs, process the input in the granular layer first, then generate an output through the Purkinje neurons, and this output is regulated by plasticity, the famous Purkinje cell plasticity, or parallel fiber Purkinje cell plasticity. Initially, it was a great discovery because they said, oh, this plasticity has to be a depression, a long-term depression. It was the time when the famous long-term potentiation of the hippocampus had been just discovered. LTP in the hippocampus was published in 1973, and long-term depression in the cerebellum in 1982. So basically, in the range of a couple of years, the two main forms of plasticity of the brain have been discovered and described, the one in the hippocampus and the other in the cerebellum, generating a lot of hypotheses and why one is potentiation, the other depressions. You know that the story evolved. The problem was not that one potentiated and one depressed. The problem is that only that these were the two forms that were discovered first. Plasticity is everywhere in the brain. It's not surprising to us that it is also in the spinal cord or wherever else. There is potentiation and depression wherever in the brain and in whatever synapse of the brain. So that was not the issue. The issue is that we have plasticity. And plasticity, now we know, is a much more complicated story than it was initially thought. It is not only in one synapse, but it is in all the synapses of the cerebellum, which changes completely the picture. And also means that when we make a model in which we have plasticity, we do not have one plasticity in one place. We have a lot of plasticity in all the places. Okay. The other thing is that in their initial theory, they didn't consider firing dynamics and molecular complexity. Why? Because they didn't know it. They simply didn't know it. They were just running ahead of time of 20, 50 years. They were really genius in their mind. But they didn't know that. So now that we have firing dynamics and molecular complexity, we are jumping on a different realm that is that of complex simulations. We need realistic models. We can't simply think about, oh, something is happening. We have to understand exactly what is happening. The other thing is geometry. This is a bit the most mysterious story, because they realized that the geometry was spatial, the geometry of connectivity, and then they didn't use it to explain the cerebellum. That's a puzzle. They used the statistics as if the cerebellum was a mesh. It was not a mesh. It's not a mesh. Geometry is strongly influenced in the way the cerebellum is computing. So all these kind of things can be summarized and explained using realistic models. That's the point. Okay, now what about, we skip all these stories. How are the neurons of the cerebellum? They're neurons, they make spikes. The simplest neuron is the granule cell. You will see that it's not so simple indeed. It makes spikes in a regular way, and not all the granule cells are regular, now we know that. And they have very different filtering properties. And also the granule cells have a resonance and oscillations in the subtracial region, which means that they are able to tune on specific input frequencies. The other neuron, the Golgi cell, is much more complicated. It's a pacemaker, makes spike frequency adaptation, post-burst pauses, is able to do post-inhibitory rebounds, and is also oscillating and resonant. Both cells oscillate and resonate in the theta band, around 4-6 Hertz in vitro. So this means these neurons are already a bit spatial. They're not just machines making spikes, not leak integrating 5 neurons, just to be clear. Yeah. In order to visualize the dynamics of the circuit, we need to go beyond the single neuron and to use techniques that allow to see many neurons simultaneously. It's a challenge. It's a challenge everywhere for every kind of every part of the brain. With the electrodes, we can't put the electrodes on every neuron. That's clear. Too many neurons, too many electrodes, too complex, impossible. What we can do is to use optical strategies, and this is an example that was developed in our laboratory, and there are also other examples elsewhere, in which the light of a laser is divided into beamlets, and these beamlets can be precisely directed, controlled, and directed on the neurons. And once they go on the neurons, they can be used to obtain information about the activity of these neurons. If the neuron contains a reporter, for example, a calcium-sensitive dye or a voltage-sensitive dye, ideally, we would like to have a voltage-sensitive dye that tells us something about the spikes. But these dye do not exist yet. So they exist, but their quantum yield is too low. So what we need to do now is to use the calcium-sensitive dyes, which tell us something about the spikes, but they don't show the spike. So we have an idea that is rather good, but finally we can get information from a lot of neurons altogether, tens or hundreds of granule cells, Golgi cells, Purkinje cells, so we can reconstruct network activity. That's an important thing, because when we speak about networks, we make a model of the network, then we need to validate and to substantiate what we model. And how we do it, we only see neurons, one by one or independent. We need also information coming from the network that is working. No? And this is what we're doing. This technique is complicated, but it's complex to use. But finally, it is possible to get information over populations of neurons. And so we use this technique as a partner of the model. This is the counterpart of the model. Okay. Now we go to the model, so the real stuff that we have to discuss here. This is an example of the granule cell. The granule cell is interesting because it is the most numerous neuron of the brain. It is the smallest neurons of the brain. It's the neurons with the less dendrites of the brain, the lowest number of dendrites of the brain. Is the neuron that is most homogeneous in the brain. Something is not true here. It's very complicated. This is an example of a reconstruction of the granule cell. With a model. The granule cell has been assumed to work as a single compartment neuron because it is so small. Electrotonically, the electrotonic length L is 0.04. It's really minuscule, very small. Consider that a pyramidal cell can have 1.5 or 2. I mean, it's very big compared. It's 1,000 times bigger, the electrotonic length of a pyramidal neuron. So in principle, we can say, well, the exchange of current from the tip of a dendrite to the soma is almost instantaneous. There is no current loss. We have estimated it experimentally. Is about 1–2%. So only 1–2% of the current is lost on the dendrite, which means it's really compact. But nevertheless, it doesn't behave as a monocompartment. Why? Because we have the axon. So the axon is a problem. The axon in many cases is neglected in simulations, or it considered just as the place where the action potential is generated. The axon is a problem. So we have given much attention to the axon, and this is a story that started in around the year 2000 and ended up last year, 2 years ago in 2016. Finally, we know how the axon, we think, we understand something more about how the axon works. The axon is made in such a way that we have an initial segment full of sodium channels that generates a large current that ignites the spike. Okay? This current backpropagates to the soma, but the soma has a sort of lower input resistance than the axon, so there is a sort of loss of current from the initial segment to the soma. And this generates a drop down of the spike. The spike is about 40–50 millivolt high here in the initial segment and goes down to 10 millivolt here. So it has a loss of 30–40 millivolts jumping from the initial segment to the soma. And this explains a puzzling observation that the action potentials of the granule cell are the smallest among those of the neurons of the brain. So the overshoot is only a couple of millivolts. But why? It was a mystery for about more than 10 years. And we did a lot of models, iterating models and experiments until we got it. So basically, there is a backpropagation that allows the spikes to become smaller in the soma, but then they have to propagate properly in the axon. That's the other point. If we have the sodium channels that are known for the brain and we put them into the initial segment, we have good spike generation, but then we do not have spike propagation. How is it possible? Because the spikes must propagate, otherwise it is not a neuron. And the problem is a bit tricky. The fact is that the sodium channel is of the kind NaV1.6, is one of the different kinds of sodium channels, and is modulated heavily by a lot of modulators. So the sodium channel, if you take the kinetic scheme of the sodium channel, this kinetic scheme will change depending on the modulators that you put on the channel. So the kinetic constants regulating the transition between states will change their value depending on the modulators that you have around. Or you can even introduce new states or kill states. So having a sodium channel, that's an important message, I think, taken from a library, is not good enough as a concept to have this channel working properly in your neuron. You have to understand what the channel is doing in reality, so you need experiments telling you what the channel is doing. In this case, the modulator is called FHF. Is the so-called fibroblast homologous factor. This fibroblast homologous factor is a factor that controls the activity of sodium channels during the growth of the neuron. At the beginning, these channels have a different inactivation properties than in the adulthood. And then the initial knowledge is that this factor only controls the growth of the neuron and the expression of channels during ontogenesis. This is not true. It continuously works in some neurons, and in this case, this factor controls the activity of the channels in the axon. So this FHF is present in the initial segment, is not present in the axon. And by doing this, it changes the inactivation properties. And you would say, yes, but how does it explain the fact that if we differentiate the channels, we have a good propagation? If we do not differentiate the channel, we have a bad propagation. The point is that the axon of the granule cell is different from the Golgi, from the Hodgkin-Huxley axon. Hodgkin and Huxley, in their model, did a great work, and they said, we have a leakage conductance. This leakage conductance is finite, as a value. And this value is not infinite. It's real and well-defined. So there is some current that goes out through the leak, always. And this generates a time constant, because the time constant of the neuron is the product of this, I talk about resistance, is the product of the resistance by the capacitance of the membrane. But if this resistance goes toward infinity, what happens there? The granule cell has a resistance in the axon that goes toward infinity. This is an experimental determination that we've done 2 years ago. And this makes a problem, because the channels are not designed to work in a membrane with quasi-infinite resistance, because all the inactivation processes and deactivation processes will be screwed up. This is easy to demonstrate. I mean, you do not have time in order to inactivate or deactivate, and the channels get stuck into the inactivated state. They never come back. The problem that if you combine this high resistance in the axon, quasi-infinite resistance in the axon, with the properties of the immature axon, of the immature sodium currents, which are conferred by the absence of the HFF factor, then you rescue the spike. And the spike travels happily at high speed without decrement, very regularly, and through the axon. So basically, the axon is something that is much more complicated than what is initially thought, and works based on a sort of tricks of biophysics and biochemistry that make the channels suitable for running in a regimen of very high resistance in the membrane. This has an implication. This spike transmission is costless, it's energyless. So you basically do not spend energy. You just need a very small gradients of sodium and very small number of channels to run the spikes through the axon. So this is the kind of things that can happen when you study carefully a neuron and you use the models. Without models, we wouldn't have understood anything of this. So having these models is fundamental and critical to understand how the neurons work. Now we try to scale up and we go toward the network. I just show a movie. These kind of neurons have been propagated into the network. And this is an example of the network of the cerebellum that is running. This is through the Lugano supercomputer. And we have a lot of granule cells, the small dots, the Golgi cells. And here you see in the center that there is something that blinks, that lights up. This is a group of granule cells that are working during an input. This input simulates what is coming from the face of a mouse. This is the simulation of a real experiment in which we stimulate the mouse, and we have the signals coming into the system, the nervous system, and going to the cerebellum. So we can simulate what is the impact and the consequence of sensory stimulation. This kind of model can be compared to the experiments again. This is an experiment in which we have taken the activity of granule cells, and this is the reconstruction of the excitator inhibitory balance in the network. This is what we get from the model. The model is able, during the same kind of input and the same kind of stimulation, is able to reproduce what the experiment were showing. So basically, we have a way to compare the model and the experiment now. This is this I would call high-level validation. Okay, so we can validate the model using experiments at high level of complexity. I don't go on the Purkinje neuron because it has been explained by Stefano. and so I skip all this part. But I jump directly to the generation of the scaffold model of the cerebellum. The scaffold model of the cerebellum is a concept that I'm not sure has been explained until now. The scaffold is a structure in which we can place neurons. The scaffold is a structure that can be modified easily, in principle, and in which the neurons can be hosted and reconnected. So when we change the neuron, we don't change all the structure, we just change the neuron, but the scaffold is still there. If we want to change a connection, we do not have to change all the network, we just change that particular connection. So the scaffold has to be a flexible tool that allows the neurons to be embedded. And there is no scaffold at the moment for the cerebellum. We're making it now. You see this is a Purkinje neuron as a flat organization of the dendrites, and these lines are all the parallel fibers. This is a picture taken from our scaffold. So now we can place the neurons, reconnect the neurons into the scaffold, and generate all the kind of networks that we would like to have. This is an example in which we have all the granule cells, a lot of red dots, the Golgi cells with Purkinje neurons, and then the molecular layer interneurons, and these are the deep cerebellar neurons, and this is the placement of neurons into the scaffold. Okay. This is activity from these neurons, and this activity resembles exactly what we have from the experiments. So we have the right sequence of activity in the different neurons, the right propagation of spikes through the networks, and this is what we can check at the beginning of our reconstruction of the cerebellar network scaffold. So now we're going beyond the granular layer, we're going beyond the input stage of the cerebellum, and we're going toward the whole cerebellum. So we're scaling up. Okay. This is the work that is programmed for the second phase of the Human Brain Project for SJ2. And this work is almost finished. We're just preparing the paper. So this scaffold is flexible, allows to host the different connection rules, including the proximity rule that is used for the hippocampus, allows to host the different neuronal model types, including single-point neurons. We do not need to have the full-blown neurons inside here, but we can. And then can run on different simulation platforms because the scaffold can be applied to PyNeuron, to PyNest, and then we already compared the performance of the two platforms using the same scaffold with simplified neurons. How do we simplify the neurons? The simplification of neurons is another problem. Everybody of you knows the leaky integrating fiber neurons that have been extended into the generalized leaky integrating fiber neurons. Now we have extended the generalized leaf, and we have constructed the EG leaf. Why we have the EG leaf? Because the EG leaf is able to reproduce many more functional states of the neuron than the G leaf, including resonance, oscillation, bursting, rebounds. The bursting was already in the G leaf, but and adaptation was obtained also in EG leaf, but all the rest was not there. The cost of this operation is that we have 3 differential equations and a couple of parameters that we have to tune. The tuning of parameters is carried out using genetic algorithms, for example, using BluePyOpt. We can easily tune all the parameters because this is a general procedure, doesn't need to work with the conductances, can work with everything. And the theory that stays behind these maths is that we have a system of equations that can nonlinear system that allow to generate oscillations in some parts of the solution space. So we have oscillations and resonance inside our equation system. We do not have to impose it. It is part of the solution. Okay. Synaptic plasticity, I already mentioned the importance of plasticity. These neurons, these simplified neurons are being used to generate networks that are embedded already into the robots in collaboration with a group in Milano, with the Politecnico di Milano. And plasticity now is placed in 3 places at the Purkinje cell synapse, in the deep cerebellar nuclei, and we're going to put it in the granule cell layer here. With this plasticity, it is possible to allow the system to get into the loops that connect the cerebellum to the rest of the brain. to run simulations and to have this kind of results in which is a bit like the movie, like Matrix. But here we have the different neurons that are running and are processing information during a learning process. Learning is autonomous. We are not telling the system what it has to learn. It simply receives inputs, has a target, and then it learns in the synapses depending on an internal optimization that is based on the synaptic rules. We are not optimizing the system. The system is optimizing itself by changing the synaptic weights. And this is exactly what the cerebellum is expected to do on the basis of theory. So we didn't impose the theory, we didn't impose the result, we simply constructed the system, so this is bottom-up in principle. We constructed the system, we allowed the system to work with internal learning rules, and the system autonomously learned how the synaptic weights have to be arranged in order to get the target. This is an autonomous system. And what we are learning out of it is how synaptic plasticity is arranged in the system and how it should or might work in the real world. Now what we have to do to go back to the experiments and see whether it is true. And this is a bit tricky. So we're trying to find out experiments in which we can demonstrate what is happening and if the prediction of the model is correct. You have already seen this picture. I don't stay on it. I just show the fact that we can reproduce Ibe-Link classical conditioning very precisely. The data have been taken from humans, and the data are distributed as the shadow gray area. And these are predictions of the model that learns to do the Ibe-Link classical conditioning task into different conditions, and it does it very precisely. So basically, the model has not been instructed to do that. It's just capable of doing it. I skip this point. So basically, the next steps are to give robots more realistic neurons, which means to transplant the new EG-leaf that we are constructing into these networks. These were already still working with the leak integrate amplifiers. So we're transplanting the new neurons. We have to have more realistic learning rules because now we have much more knowledge on the learning rules than we had when we did these experiments 2 years ago. So we can transplant and improve and scale up the learning rules. And then I just conclude with the last picture, the big problem. How to connect the knowledge that we get from the integrated signals with the knowledge that we have from the bottom-up models and from the experiments done in the lab. So this is what normally happens. This is how many people see the brain, no? Because here you really see something, you see spots, and you say, now I understand how the brain works. Well, more or less. I mean, the point here is that each one of these spots means that you have millions and millions of neurons running under the spot. And what are they doing? So I can tell you what the neurons can do starting from my models. But I can't tell you what the neurons are doing here. So how to connect these two worlds? This world is very important because medicine is running here. The diagnosis of pathologies is running here, not with our models. So we have to reconnect the two things if we want that our work is useful to understand what is happening here. So the point can be summarized in this way. We have to find the convergence of this kind of measurements that start from here and go down toward this kind of measurements going here. So we converge where? Basically in the microcircuits. This is the point of convergence. So we have exactly in our hands a key that could in the future be used to understand what happens in these signals. For example, for the cerebellum, we're in a good position because we have these experiments. We're doing them, actually. We have the models, and we have also the cellular experiments. So we can really have a convergence point of all these approaches. This is something that they left open, and we're making projects on it. And we're trying to find a way to put these things together. It's a complex story, but in principle is feasible. I have to think clearly I don't do all the story alone. There are many people working. And, well, thanks to everybody working in this big project. Thank you.